Good morning. There's the high loves with a sudden stop. And I hope you can hear me. Today we're talking about hearing. We're talking about mechanoreception, which can be other things other than strictly hearing. But hearing is going to be a lot of the examples that we talk about today. So I don't know if you found any of the letters and unscrambled it, but you can zoom back to that to take a look at it if you're interested. This is the last of our sections in the kind of the chapter on sensory systems and the diversity of sensory systems that exist across the diversity of animal life. That is the diversity of the phyletic group that we love, the invertebrates. Previously, we'd been talking about chemoreception, the smellers and the tasters. Today, we're going to be talking about things that listen. We're going to sit down and we're going to listen. We're going to talk about mechanoreception and proprioreception and kind of so the response to knocking and vibrations. We'll talk in the marine example about statocysts. We'll talk about halteres in insects, slit sensilla in spiders. Ants are going to feature again as some of the examples are there, how they can differentiate between the vibrations of winds and herbivores. And then we'll kind of focus at the end thinking about examples of acoustic communication, the problems with sound production or like challenges associated with sound production and sound detection. And we'll go into some specific examples with ants and with katydids and parasitoids. That is the plan for where we're going. So my hopes for all of us today is that you, come on, fly. Those little tiny halteres beating behind the wings. That you can appraise, synthesize, and interpret any example that you bump into in your life from now on about mechanoreception and the diversity of strategies that invertebrates have for dealing with that. And then think about how it's conducted or how it's manipulated by predators and prey in the larger world of through ecological time and into evolutionary time. So again, back to The Simpsons, when we're talking about the modalities, we dealt last week, a week ago today, with vision and photoreception. On Monday, we talked about taste and smell, olfactory and gustatory with the chemoreceptors. And so today we're talking about touch and hearing. And when we're thinking about this, in the in the invertebrate world, bigger than the insect world, but here with an insect or invertebrate example, an insect example, this fly leg, where in green you can see there's a number of different mechanoreceptors. They have a hair plate at the coxa, which is nestled right up against the body. They have a campaniform sensilla, which are these kind of two little drum skins there. They have a chorodontal organ stretched across a join in a leg, and then they have hair bristles on the outside. So the chorodontal organ is inside, the bristles are stretched across the outside. And so whenever we're talking about like the diversity of strategies on the fly leg or zooming into what some of these bristles would look like and how they're connected to the nervous system, a bristle extending out of the body, a hair plate, there's the little dendrite and the campaniform sensillum, and then zooming in at the chorodontal organ. Or with the statocyst, which is not imaged here. What we're talking about is things that are being deflected, tension that is being created or resolved, stretching or compression leading to any of those organs that connects to the firing of the of neurons that are connected into the sensory system that allow and permit the organism to interact with its environment. To interact for what? Well, to interact so that it can move safely, to interact so that it can court and engage in sexual relations with others of the same species. That stepping back from that there can just be conspecific recognition, that there can be predation, that there can be the defense against predation, or there can just be kind of subtle eavesdropping on what's going on around the world, around you, around it, and that particular day. So there's our first reminder from Caramel. I don't see any questions as of yet. So we're talking about organs that detect this mechanical kind of stimulus. So touch, pressure changes, vibration, and sound. And of course, those are not independent categories, right? So when we're talking about sound, we're talking about changes in vibration, in pressure that are received by different organs, different hairs, different statocysts, different tympanum, those kinds of things. So the bottom example with this hair, we have a sensory receptor rooting through into the nervous system and then extending out into and above the cuticle. Okay, so this is flexible. So this bristle or this hair at the end of the last lecture, we talked about Brome's bristles that are at the antennal segments of all the arthropods. So it's a flexible cuticle, so it can respond to touch or pressure, vibrations, or sound. And so the bending of there is then transmitted into the sensory system. You can also have this deformable cuticular plate. That's more like the campaniform that we talked about, I used to have one right here. It's to demonstrate a lid, like a lid off of a spaghetti or any kind of a jar. And let's start out though with the statolith, because the statolith is one of the most widespread from an evolutionary perspective, from a phylogenetic perspective, and even from a global perspective, because it's found in jellies. And remember, jellies are found all across the world and all across, if your Elio is going to land, if the alien is going to land in the planet Earth, they're going to wonder why it's called Earth, because it seems to be predominantly water and is predominantly peopled by jelly. So statocysts are found particularly in pelagic forms where geotaxis is an obvious advantage. And so what we have here is a diagram of a jellyfish statolith. So there's this kind of a dense ball that's maintained in a circle of fluid that has sensory, into which are project sensory cilia. Okay, and so as the dense particles of the statolith kind of move and bounce around, they bump into the cilia, and then they provide feedback to the organism about which way it's tipping. Think of it of the little ball of air, so that not a ball of solid, but a ball of air in your level as you're setting up your bookshelf or your pictures, and you're using that kind of eyeball. You're using that ball of air inside the level to do that. And so invertebrates across, so there's a bunch of different taxia. We've got lots of gastropod examples. We have a bivalve example. We have a tunicate example. These line diagrams on the left-hand side are just picturing some of the range of statoliths. So you see lots of them fit this kind of classic form that we looked at in the last kind of greeny tinge example. But we have the cilia projecting into the interior of the ball of the fluid-filled area, where the statolith kind of bounces around, and what it's touching on the inside provides the feedback to the organism about geotaxis, or which way is up, that kind of thing. But it can be, the ball can change shape, the density of the cilia can change shape, the length of the cilia can change shape, and in fact, it can operate more like a snow globe or an hourglass, in terms of some of them can just be particles, not just one dense ball that's moving around, but just particles that are kind of falling around inside of that sac. And this is used these are all adult pictures here, or pictures derived from adults, this kind of mineralized mass with the associated cilia. But it's also used and quite important for larval stages, and particularly for planktotrophic stages that may settle out eventually, or lectotrophic stages that settle out of the water column eventually and no longer need their statolith so much if they become rooted, but when they're part of the plankton, they need to be able to respond to up and down because that's generally associated with how they are feeding. So here's the return to that green diagram of the jellyfish, and you can see this is on a cubozoan, and here's a zooming in on this. So last year when we were talking about cnidarians, we talked about the sense organs, the rhopalia, that appear here and here at the edges, just on the bell margin. And so that's a statocyst, right? And it's involved in this case for a lot of things. It's a multi-purpose sensory tool for this particular lineage of cnidarians, but it's, from our perspective today, this is a mechanoreceptor, a sense organ that is responding to the world. So you have this fluid-filled cavity with the mineralized mass that's kind of bouncing around, and there's on the edge, we've got sensory epithelium. And you can see it inside of diverse taxa, from crayfish to octopus. And so there's, on the bottom here, there's your crayfish example, that it can be a dense mass, or it can be free-moving and loosely attached smaller granules. So we've talked about bivalves, gastropods, tunicates, decapods, different lineages of mollusk, and in fact even into the ctenophores. So this is looking at one of my favorite lineages. You've got ctenophores with this kind of gaping mouth here at the bottom, and the biggest thing that's moving around simply by ciliary motion, the ctene rows on the outside. But at the back end, by the transient anus, if you remember the ctenophore time we had together last year, you have the statocyst. So this sphere in which this mineralized mass kind of bounces around into cilia that you wouldn't quite be able to perceive in this light illuminated video here on the bottom. And so that can translate into, those signals are transferred into the nervous system and out into the ctene rows to coordinate and to allow kind of forwards and backwards movement so that it can move around in relation to any particular vibration or other of a prey item, for instance. Remember, these are great big predators. So there's a diagram just showing that the granules kind of shifting around in response to gravity, and allowing the translation into specific movements by that gorgeous ctenophore. And other taxa that I love a whole bunch, we remember the purple sock and Xenoturbella. They also have a statolith. They have practically nothing else. Or if you remember the simplification of this earliest branching out, or the bilaterian, they've lost most of the other characteristics, synapomorphies of being a bilaterian, but we have a mouth, and we have a really simple body, but at not associated, so the mouth is on the bottom, but maybe let's not even go there. Let's not call it the anterior, but at one end of the body, we see statolith. And that's what it would look like under light microscopy. And if you're a taxonomist of Xenoturbella and you dissect them out, you can see a section here, because they've survived histology, and they're thinly slicing through the anterior end, or that end of the Xenoturbella, and they've captured several different kind of bouncing around statolith and ciliary hairs. So in this case, the cilia aren't on the edge of the vesicle that's containing the liquid, but it's actually attached to, almost like a little protozoan here. The statolith has its own cilia, and they're bouncing around with a heavy end and another end that's delivering information to the organism. If you were to zoom inside that crayfish head, you could see the statolith here that's been cut away on the base of this antenna. And it's a balance organ for lots of crustaceans that's in the basal article, so the first kind of segment, the one that's closest to the head. And we've talked a number of times about the problems associated with being big, problems associated with molting, and we've mentioned that it's from an arthropod, for an insect perspective, from a crustacean perspective, it means you can't breathe. We didn't call attention to the fact that this basal article that contains your statolith is also replaced every molt. So not only are you not as hard and defensively oriented against predators, not only can you not respire as well, you can't even balance that well, because your statolith at the base of your antenna has been replaced, and it needs to harden and reform every time that you grow. When we talk about sound, when we talk about response to vibrations, some of you are maybe more have a greater intuition about this than others in terms of how sensitive you might be to sounds. But I'm going to ask a very simple question on a marinescape scale, on a big scale, and say, is sound a stressful environment? And how would you measure it? So researchers here have measured for a particular species of crab whether or not a quiet or a loud environment is stressful. And so what I'd like you to do is to grab your annotation tool and think about showing me what it would look like. You can choose red or blue, but show me what it would look like just basically across the x-axis, which is size. How you would expect oxygen consumption to scale. What kind of a relationship ought there to be between size and oxygen consumption? And then, with a different color or a different line, show me what it would look like if it was in a loud environment that was stressful. Awesome. Okay, throw in the chat, interpret some of these for me, because the colors, so we've got a normal environment, I think largely in red, and then a bluer or a louder environment in blue. Is that, have you followed the color scheme? I'm guessing. Amazing, thank you. Okay, this is awesome. Well done. Put your hand up in the air, pat yourself on the head. This is pretty much exactly what we'd expect to see. That is, bigger, if things get bigger, they ought to, metabolically, they ought to consume more oxygen, right? Now, a stressed organism should likely consume even more oxygen on top of that. And that's what you're sketching out here, predicting that either the intercept and or the slope of this relationship between body sizes and oxygen consumption is going to change. I'm going to take away this so that you can see kind of exactly what they found. Without the great colors, pretty much exactly what you predicted. Both the intercept and the slope of that relationship would change. This is support for the fact that a noisy environment is a stressful environment. And this is particularly important. This is another signature of the Anthropocene, that in marine and aquatic systems, we make our activities, our human activities, make a lot of noise. And this can be harmful and stressful. All right, there is Caramel. I don't see any questions. Let's move into the terrestrial environment a little bit, and we'll think about a dipteran example. So, within flies, so true flies, Diptera, and if you remember back to thinking about them, or if you're passionate about flies in general, you know that the derivation of that word, these are two-winged. So instead of having four wings, we see writ large that there are two wings. And so on this fly right here, you can see the second set of wings have been dramatically reduced in true flies, and they've become these kind of modified clubs or halteres. So the hind wings, and there's just an aside to another order of insect, the strepsitrons, which are weird in a whole bunch of ways. But in their case, they've also had a reduction in wings, and in their case, it's the other ones. It's the fore wings, not the hind wings. So in true flies, the hind wing has been reduced into what's called a haltere. Or a haltere. And so inside of that yellow diagram, so this is the same kind of pinned fly that is glued at the base, and we're filming it in high resolution in slow motion. And I hope inside you can see two little white clubs kind of moving around. These are halteres that allow the fly, and they're to move really precisely. They're essentially acting as a strain gauge because of different bristles that are retained on this little club, this reduced wing, which allow them to act as kind of controls of equilibrium as they're flying. So they can balance and they can guide and provide rapid feedback to the muscles that are steering the wing, and then therefore the head, so that they can either engage in, you remember the robber fly that was kind of zooming out and eating very, very efficiently, and doing those kind of guided empathetic movements in relation to its prey. Despite the fact that it has a tiny, tiny, tiny brain, it's responding to the prey. So that's a mixture then, in that robber fly example, of photoreception, that complicated eye, into the tiny brain, but also the halteres that are allowing them to keep the head stable and also do the minute controls of speed and direction that allow them to hawk out and intercept their prey item. Let's take a look in at these, zoom in, and have a look at the drumstick of this fly. So this is the haltere. So you can see these papillae and these plates, so campaniforms and Hicks papillae. These are all of the kind of the little sense organs that are on that beating haltere. Now, the halteres tend to beat with the same frequency as the forewings, but they can be decoupled. And it happens in kind of a cool way. So the haltere in this diagram really simple, almost like a circuitry diagram on the bottom. We've got the halteres now, the drumsticks in red, and they've got their own set of muscles, but then there is a link between the haltere, the former hind wing, through the scutellum and into the connection points for the forewing. So what's doing a lot of the motion. Now, what's been talked about, or the way that entomologists and morphologists within the study of insects have talked about this, is essentially like a gearbox, that they can be coupled directly so that the beating of the haltere is in exact synchrony with the forewing, or they can be uncoupled depending on whether or not this clutch, this serration in the cuticle, is the teeth are engaged or not engaged, just as you would in your car. And so we've got a disengaged state here, and then an engaged state in the bottom. So that coupling might be important depending on how, in response to, like I said, as a gyroscope to maintain kind of position in its mating flight, but also because there's, of course, what do you call a fly with no wings? It's a walk. Flies walk sometimes, and so perhaps you don't want the halteres. You still want the information that the halteres are giving you, but you don't want them to be in synchrony with your wings if what you're doing is walking through the leaf litter. So disengaging, decoupling this clutch, releasing the clutch is a really important thing. We're going to switch taxa within the arthropods. We talked a little bit about spiders the other day when we talked about the wall pore cincillae, and you showed me that they were all over the upper parts of the legs of male spiders, not on females, not on immature males, and not on the distal ends that would be kind of like doing all the hard work at climbing. Now, spiders are full, and they have we talked about that from the perspective of sexual communication when you don't have antenna, because we've been spending a lot of time in the chemoreceptory field thinking about antenna. Now, spiders are full of mechanoreceptors. And if you think about just a classic kind of a Charlotte's Web kind of a world, or a big orb weaving spider, vibrations are really critical, not just for hearing when the big lumbering primate is coming through the bush and might kind of break through your gorgeous web, but also to understand when your web has captured a prey item, and to understand how big it is and whether or not it's worth just kind of cutting it loose because you might get damaged yourself, or if it's a perfect kind of a prey item that you wish to go in there and release and tidy up and begin to eat. So if you had the opportunity to look through very, very closely at a spider leg, you would see all of these different kind of amazing sense organs that are, some of them hygroreceptors, so responding to like this pit organ here that's responding to humidity, or this the taste sensitive claw right at the end here, darker gray. But so many of these are like the trichobathyphrium, or the trichobathyphria, which are for sensing air movement and low frequency sound, or slit organs that are for sensing vibrations and limb positions. So that slit organ right there, let's talk about that, because we've kind of talked about some hairs or cilia for a little while, but let's think about basically a strain gauge. It says here, so a slit organ is like a strain gauge. So it's right here, and we're going to zoom in the next slide. You can see it's there. It looks like that accordion-like feature there. So what we're looking at is essentially kind of the rolled up section, deep grooves in the cuticle that are associated and rooted in sensory in neurons that can then respond to differences in pressure. They can be, if they're at leg joints, they can be proprioreceptors so that the spider can feel how roughly or strongly it's holding on to or touching something. Georeceptors, mechanoreceptors to external pressure, vibration receptors, or phonoreceptors, so actually to hear. And so we've got some SEM here. There, we've got the slit cincilla and the compound organ here, or the lycoform on the trochanter, so different on different sections of the legs. And these are really important for understanding changes in pressure that are translated into the hemolymph, into the nervous system, and then into the central nervous system and back into muscles. We, as our species, as we've learned more about these kinds of sense organs, we've biomimetically, we've used them. We've made some architectural advantages based on understanding these slit sensilla of spiders. So we've got slit sensilla here. So there's the slit organ, and you can see the cuticle kind of all bent up there. So sorry, going the wrong way. The deep grooves in the cuticle. Now, so as that is pressured, it's a proprioceptor, but it can also respond to all sorts of things like we talked, and it's rooted in the sensory neuron. So that's the spider story, right, that we just covered. But what different engineers have done is adapted the slit sensilla and used it in building materials, particularly in large-scale building materials in areas that are prone to shaking and to earthquakes. And so they've built in these small cracks that can then shift load and respond to different vibrations, different movements in the environment. And not just crack, but just shift its load and respond. Now, if we think about shifting and responding to an environment, this is our first ant example. These are acacia ants. Acacia ants, which we've talked about briefly a couple times before. These are ants that live the entirety of their life on an acacia tree. So it's a species or a couple of different genera that have done it a couple of different times in 2 different continents. This is an African example. So it's a true acacia tree that provides for the ant. It's very mutual. It's a classic mutualism. So the tree provides for the ant food and housing, and the ant provides for the plant defense against herbivory. The idea is that the ants live in the swollen bulbs on these thorns and come out and they sting and bite ferociously any herbivore like this goat that might be browsing on it, which leads to a question, well, there's all sorts of vibration environments when you're a bush, right? This isn't a great big solid tree. It's a small tree. It's a bush. So it's going to move around in the wind. And how do the ants not waste their energy defending against the wind? How do they only go out and respond? Or can they? Are they sometimes with ants, they're so numerous we don't expect to see perfect behavior. We expect to see kind of good enough behavior. So do they waste their time? And we know now that ants actually do spend lots of their time wasting. But in this case, can they differentiate between herbivory and wind? And what they found with visual surveys was that the ants didn't increase their patrolling behavior in response to wind-induced vibrations, but that the number of patrolling ants did increase immediately, as you would have expected, as they expected, for browsing-induced vibrations. When they put in the goat, the ants came out. And so what they found was that there were, when they measured the vibrations, the environment, how these movements are being transferred through the material of the plant, what they found here was that wind was biased towards lower frequencies, but when it was a herbivore, there were higher frequency components, up to 6 kilohertz here, that was the symptom that the ants were responding to and identifying through different bristles and different mechanoreceptors on their body and on their antenna to say this is not the wind, this is now also, or instead, a goat. Okie doke. There's Caramel. Let's switch for the rest of the time to thinking about sound. When we think about sound, so not just vibrations, not just geotaxis, when we think about sound, particularly in a terrestrial environment now, there's some advantages towards communicating with conspecifics or in a sexual perspective via sound. And if you've got the equipment that you can interpret the vibrations that are moving through the air. It's not limited so much by environmental barriers. I don't need to be able to see you. It can kind of transmit through that. It does pretty well across larger distances and around corners. And there's what you would call high information content, that it can be very variable, highly variable, and it can change quickly. Disadvantages include the fact that individuals and non-conspecifics that you don't intend to receive your signal can use it, can eavesdrop on it, and can track you down. So predators can track their prey by the sounds that they produce. It's less effective the noisier the environment gets. We already talked about that with a marine perspective, but of course in a terrestrial perspective, acoustic communication is really hampered in the Anthropocene, especially in urban areas, because we make a lot of noise. It can also be metabolically expensive to produce, especially if it's going to and maintain. If it involves specialized structures that maybe have their own pros and cons, or cons essentially in this case about that prevent you from hiding or prevent you from doing something else. Now, let's think about, basically, when we're thinking about sound production, let's think about different ways that it can be produced. You can stridulate, you can rub one body part against another, especially across teeth. You can vibrate a membrane, you can knock one's body against the surface, you can push air out spiracles, or you can vibrate your wings. This is
such
a cicada example that we're going to spend 20 seconds with.
Such high volume is through its hollow abdomen. It's got these little timbals, and it pulls in and out these drum-like organs.
So this is a vibration.
And with the hollow abdomen, it's able to create a huge amount of noise.
And amplify that sound, and then send it farther and be that much more attractive to any particular conspecific that you're trying to attract to you. Sound detection, we've got a bunch of different ways, and they are largely some of the same organs that we've talked about. You can have antennal hairs like on the antenna, like this arista. You can have a tympanic membrane. You can have bristles or choriodontal organs, as we talked about on the fly. So production and detection. Sometimes a tympanum, sometimes serration is involved in either. Let's take a look at a cricket, a famous example for making sound. So we've got that chirping, where if you were to look at, as you watch the video, the wings are moving, and they're moving. There's a top one, and if you flip it over and take a look at this region right here, what you'll find is that there's a vein along the upper wing that is covered in serrations, and as each one of those teeth rubs against the other wing, it produces an oscillation. And then grouped together, all those teeth together forms this pulse that creates, that you see kind of marching across the screen there. So that's the chirping cricket. How is the cricket receiving? Well, in the front legs, they have a small tympanic membrane that vibrates in response to the sound. And the amplitude of that vibration is greatest for frequencies that are near the fundamental frequency, the characteristic frequency for the species that is calling. Now, those sonograms, I'm going to show you some videos here that I've taken from a cricket researcher's presentation that you can go and watch later on. It's kind of fascinating. But it calls attention to the fact that this sonic environment has been very fertile in terms of communication, particularly for many orthopterans, like this particular group of insects, terrestrial insect. But also these sounds are coexisting in a complicated environment. And it's coexisting because what's being produced the sound that's being produced is very close to the frequencies that the ear of that species is best at hearing. It's better at hearing conspecifics than others. We're going to see a slow motion video of that same chirp and the same serrations here.
Video. So this is slow motion video. Now.
So those are the serrations across, and then if we were to see perhaps an environment that has a bunch of or a number of different species.
Video. These are all of them singing at night in the ultrasound.
So whistles, clicks,
That's how the ultrasound treat the baby.
vibrations, a complicated environment. And so that tympanum is tuned essentially to that sonic frequency, to that sonogram of what the male is producing. And that ear is then innervated heavily, 60 to 70 different receptor neurons. Thinking about stridulation, or so about the serrations, crickets are a classic example that we would be kind of impoverished if we didn't consider, but we would also be impoverished if we didn't kind of talk about the fact that ants also use this. I think this is pretty cool. So this is an ant that I grew up calling in Costa Rica Pachycondyla apicalis, that has now been reconfigured as Neoponera apicalis, as the borders of this genus were kind of reconfigured. It was what taxonomists would call a trash can of a genus, because largely it was, especially standing up and looking down at the ground, it was apparent. It has these kind of bright yellow tips to its antenna. It's a large thing that you can see without a microscope. It's like, oh yeah, there's Pachycondyla, or now Neoponera apicalis. But what we have found is that there seem to be multiple different species masquerading within that one name. And they communicate differently, not just with chemoreception, because certainly they do. The cuticular hydrocarbons are different, yada yada. But also, if you look at this segment, the interface of these two abdominal segments, and you see this tiny region, this is full of serrations that the ant can then pump in and out and make calls. They can talk to each other. They're not just talking. So sight, not so important, but sound and smell for ants, critically important. And so for different prospective species of Pachycondyla apicalis, if we look at the serrations on that abdominal segment, we hear them. They make different sounds. Versus so this combination of essentially it's integrative taxonomy in the same way that lots of bird work is done. We're looking at not just how they look, but how they speak to each other. We're listening to the ant calls and then using genetics as well to kind of go into a large mass to say this is not variation within a species. These are different things. And it makes me think most ant species have many other Myrmecophilus, so species that live within their colony. And you think about adaptations that those species would have to not be immediately set upon and eaten as prey, both chemoreceptory and potentially mechanoreceptory adaptations of the species of beetle or whatever it is that's living inside of that ant colony. Do they speak ant? Do they smell like ants? One of the last examples here, last final two, is we've got a classic one. We're back into the orthopterans, into katydids, or these bush crickets, because they have kind of incredible organs on their knees. They have a tympanum on their knees, which is acting as an ear. So you see, and this is kind of a dissected away. So this is a dead individual where we have on the knee this tympanal organ that is connected via a long trachea, an ear canal, that is then has an acoustic spiracle at the end. So behind the tympanum, an acoustic trachea that runs along internally to the leg and then opens out into the body. And so these eardrums on each leg essentially receive sound from the outside, but also a slightly delayed sound internally via this tracheal tube. And what researchers have figured out is that this tube, this acoustic vesicle, which is shown in a rainbow way here, that is colored, the rainbow is the changing in decibels and sound pressure along the tube. So there's the eardrum here, and it's being affected like from your perspective, from the side. But it's secondarily and more slowly being fed information from the tympanal organ that's moving along this hollow fluid-filled tube, and it's being amplified. So it's receiving perhaps more subtle information, but certainly information that, because it's being received by the central nervous system at two different stages, that's allowing really fine-grained response to that environmental cue. And so here, what we're looking at is this stylized diagram of scientists being asked, well, what would it look like if you move through this acoustic vesicle? It would look like this. So you are now a sound, a wave propagating through the acoustic vessel of this bush cricket, Copophora gorgonensis. So they're hearing it so 4 times. How does that work out? So 2 different sides of the body, 2 different sets of knees, 4 different ways of interpreting that sound. In this species, it's nocturnal, and the males are singing to attract distant females for mating. So that's clearly being able to respond to sounds like that is involved in that. However, they are a species that is also preyed upon by bats. And by comparing, and bats using ultrasound is potentially one way. Another way that having these 4 different inputs and slightly delayed inputs would allow Copophora gorgonensis both to speak to each other, to conspecifics, and also to understand and to avoid when bats are around. So how do they do that? Well, here researchers have looked at is the pinnae, which is kind of like we've got the tympanum, that stretched membrane, and there's a ring around the membrane. So it's a membrane on top of a membrane. And this is the pinnae. And they don't seem to assist in the calling of interpreting conspecific calls. So this is the gain of a left and right, an anterior and a posterior in this case on one. So one knee. So not left and right, but on the anterior side of the tympanum and the posterior side. And you can see there's a big frequency gain in the higher frequencies. So like above 100, there's a peak there, I don't know, 60, 100 and 120. So that's not helping them here. This is where conspecifics call. What that's doing is acting as an ultrasound detector. These are the 3 principal bat species that live around this population of gorgonensis. And so what it's showing is that this is essentially the tool that these have, the tympanum, the pinnae on a tympanum, the membrane on a membrane, on the knee, that allows the cricket, the katydid in this case, to speak to each other, but also to avoid being predated by bats. So when we see tympanum, we think sound production, we think sound reception and interception. But if you were watching the news last week, with some of the more biologically oriented news, what you saw was that there was a species that was described where the mature female has a tympanum on its leg that we have. So the males don't have it, the females have it on the hind leg. And it was always interpreted as a sound, just like that gorgonensis example, that this was, the females had it because they were listening to the calling male. It made some intuitive sense. But what some close examinations has found is that this cuticle is not just a tight drum, that there's a whole bunch of different pores and holes, and it has the consistency that a drum skin would never have. And when they grew some of the material on that, what they found was that there was a bunch of fungal hyphae that were being grown there. That they essentially had this pit in which they're growing a fungus, and when they lay their eggs, they rub the cultivated hyphae across the egg mass. And then those hyphae, so the eggs are laid, they're covered in the hyphae from the fungus that the female has been growing in the pit in her arm or her leg, and that grows over top of the eggs and then prevents parasitoids. So kind of amazing. So tympanum's always a tympanum unless it's the sac in which you grow the mushrooms that you protect your eggs with in the future. Last example here. Again, it's with sound, and it's with a parasitoid, but it's an adaptation, a kind of a clutch or a coordination example that a particular parasitoid fly uses. So this is an ingenious little trap that a researcher has used. These are the parasitoids are the flies you see moving around. And you see right here under my cursor, there's a break. Inside, there's a box underneath where there is a recording of a species of cricket being played. And so the flies are coming in to find and parasitize that cricket. Now what the researcher wants the flies, because the researcher is going to study the flies and how they're using auditory cues, how the female parasitoid is using the auditory cues, eavesdropping essentially on the auditory cues of the male cricket. so that it can produce its own eggs. And what they found is that inside of this fly's ear, so the eardrum here right in the central segment, has a pivot point on both sides of the body. So we're looking at the fly, and you're kind of zooming in and looking from this post on Blue Sky from a famous fly taxonomist at the Natural History Museum in the UK, from a fly that she collected in Honduras a few years ago. But they have this pivot point with these two hearing organs on either side. So there's a bilateral nature to so thinking about directionality, that's clearly going to be important here. But it's also important because the fly is much smaller than the cricket. And when we think about those sonograms, essentially what you're looking at is at its size of the movement of that sound, of that vibration through the air. And because of the small size of the fly and the necessarily even smaller size of their ears, relative to the wavelength of the cricket sound, their directional cues are restricted, except they have this fixed support, this pivot point, this intertympanal bridge. So the head has been dissected off this unfortunate fly with line diagrams to show you kind of where it sits. So the tympanum is here, and we have two different membranes on either side, these pits on either side across a bridge. So that coupling between the two tympanal membranes allows the amplification of small direction independent vibrations in the field. And this allows rapid integration of that vibratory environment so that the fly can track down the calling cricket in a way that it couldn't otherwise. So we've got the tympanal membrane on either. Here's an even more simplified diagram from the bisected head or the dissected head. But we've got the pivot point or the bridge. So we've got a scenario where there's symmetric vibration where it's probably dead in front of you to it's a little bit off, left greater than right, right greater than left. And so that allows the direction and of minuscule interaural, so between those ears time, and the use of this information-rich acoustic directional signal. And just like the cincilla or the grooves and the cuticle of the spider being used biomimetically by engineers, we have followed up on this, and there are all sorts of acoustic microsensor. There's a whole industry of people trying to adapt what this particular species of parasitoid fly, how it hears, so to improve our sub-wavelength kind of directional hearing and essentially adapt this rocking model, so the bridge with the two free tympano on either side. So micro electromechanical acoustic sensors, the inspiring environment of the invertebrate world. All right. This is it. This is the end of our sensory system. We've talked about staticists and halteres. We've talked about flies a couple of different times, parasitic and non-parasitic, ants a couple of times, and some orthopteran examples with the bush cricket and with calling or chirping crickets. Where are we going? Next week, we're going to enter into some time where we're going to be thinking about movement. We're going to spend Monday talking about flying, and then on Wednesday we'll talk about some other kinds of some swimming, crawling, walking, and running. What else should you be working on? Your lab report is due next week. Your InvertR assignment number 2 is due this Friday. Your case study is available as it is every week from Thursday morning to Monday morning. And I'm going to be incommunicado on Saturday, I believe, traveling again, so I won't be able to respond quickly. But I hope this has been enjoyable, and take care of yourself in the meantime. Take care of others around you. I'm going to stop recording here in a second, and I'll hang out. If there's any questions, I will try and answer them. But until then, take care.